Have you ever wondered what really happens inside your body at the smallest level? Inside every cell, there is an entire world working non stop to keep you alive. From tiny atoms to powerful energy systems, today we are going on a journey into the chemistry of life itself. Welcome to Deep Insight, and this is Introduction to Biochemistry. Everything begins with atoms, the smallest units of matter. Carbon, hydrogen, oxygen, nitrogen, these are the building blocks of life. But atoms alone cannot create life, they must connect. They form bonds like friendships. Some atoms share electrons called covalent bonds, others transfer electrons called ionic bonds. And when these atoms join together, they form molecules, the foundation of life. Molecules become more complex, forming biomolecules. These are the real heroes inside you. Carbohydrates gives you energy, lipids stores energy and builds membranes, proteins do almost everything like structure, movement, reactions, etc. Nucleic acids like DNA store your genetic information and vitamins help all processes run smoothly. Think of them as a team, each with a unique role, working together to keep you alive. Have you ever wondered where your body gets the energy to run, think, and even breathe? The answer lies in carbohydrates, one of the most important biomolecule in our body. Carbohydrates are organic compounds made up of carbon, hydrogen, and oxygen with a general formula of CH2ON. They are the main source of energy for all living organisms. When you eat foods like rice, bread, or even fruits, your body breaks down them into glucose. This glucose is then converted into ATP, the energy currency of the cell, powering every function in our body. Carbohydrates are of three types. Monosaccharides, like glucose and fructose, are the simplest and easy absorbed. Disaccharides, like sucrose and lactose, contains two sugar units. And polysaccharides, like starch, glycogen, and cellulose, are complex and used for storage and structure. In simple terms, carbohydrates are polyhydroxyaldehydes or ketones, or substances that form them when broken down. So, next time when you eat, remember you are fueling your body with energy at the molecular level. Have you ever wondered how your body stores energy for a long time or how it protects your organs and keep you warm? The answer lies in lipids, another essential group of biomolecules. Lipids are organic compounds mainly made up of carbon, hydrogen, and oxygen, and sometimes phosphorus and nitrogen. Unlike carbohydrates, lipids do not have a fixed formula and they are hydrophobic, meaning they do not mix with water. That's why oil and water never mix. Lipids play a very important role in our body. They store long term energy even more than carbohydrates. They protect vital organs, acting as shock absorbers, as they help maintain body temperature by providing insulation. Lipids are also key components of cell membranes, especially phospholipids. There are three main types of lipids: simple lipids like fats and oils used for energy storage, compound lipids like phospholipids, which help for cell membrane, and derived lipids like steroids, including cholesterol and hormones. When you eat foods like butter, oil, nuts, or ghee, your body stores this energy as fat and uses it when needed. In simple terms, lipids are hydrophobic organic compounds, insoluble in water and soluble in nonpolar solvents. So remember, lipids are your body's long term energy reserve and protection system. Have you ever wondered what builds your muscles, repairs your body, and helps every reaction happens inside you? The answer lies in proteins, one of the most important biomolecules of life. Proteins are organic compounds made of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. They are built from smaller units called amino acids, joined together by peptide bonds. When these amino acids fold into specific shape, they form functional proteins. Proteins play many vital roles in the body. They help in the bodybuilding and tissue repair like muscles. They act as enzymes, speeding up chemical reactions. They function as hormones like insulin, and they support immunity through antibodies. Proteins also help in transport like hemoglobin, carry oxygen in blood. There are two types of protein: structural protein, which provides strength and support, and functional protein, like enzymes, hormones, and transport proteins. When you eat food like eggs, meat, pulses, and milk, your body breaks them into amino acids and uses them to build new proteins. In simple terms, proteins are polymers of amino acids linked by peptide bonds. So remember, proteins are the building blocks, workers, and protection of your body. Have you ever wondered how your body knows how to grow, look like your parents, and control every function inside you? The answer is nucleic acids, special biomolecule that stores and transfer genetic information. Nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They play a key role in controlling all cells' activities and are essential for life and inheritance. There are two main types of nucleic acids: DNA or deoxyribonucleic acid, which stores genetic information, and RNA or ribonucleic acid, which helps in protein synthesis. The basic unit of nucleic acid is the nucleotide. Each nucleotide has three parts: a nitrogenous base, a sugar, and a phosphate group. These nucleotides join together to form long chains. In RNA, there are three important types: mRNA carries instructions from DNA, tRNA brings amino acids, and rRNA from ribosomes where proteins are made. This flow of information from DNA to RNA to proteins is called the central dogma of biology. In simple terms, nucleic acids are biomolecules that store, transmit, and express genetic information. So remember, nucleic acids are the blueprint of life. Have you ever wondered why eating fruits and vegetables keep you healthy, boost your immunity, and help your body function smoothly? The answer lies in vitamin. Essential biomolecules required in very small amounts. Vitamins help regulate body function, and although they do not provide energy directly, they are crucial for metabolism. That's why they are called micronutrients. Vitamins have some key characteristics. They are needed in small quantities, and most cannot be synthesized by the body. So we must obtain them from food. They are essential for growth, immunity, and overall health. There are two main types of vitamins: water soluble vitamins like vitamin B complex and vitamin C, which dissolves in water and needed regular intake, and fat soluble vitamins like vitamin A, vitamin D, and vitamin K, which can be stored in the body. Vitamin plays many important roles. They boost immunity, maintain vision, support bone health, help metabolism, and aid in clotting and skin health. When you eat a balanced diet with fruits, vegetables, milk, and nuts, your body gets all the vitamins it needed. But lack of vitamins can lead to deficiency diseases and weak immunity. In simple terms, vitamins are essential micronutrients that regulate body function and keep you healthy. We have learned about all the five types of biomolecules. But what holds all these molecules together? They are invisible forces. Invisible forces like hydrogen bonds, which are weak but essential, hydrophobic interaction, which keeps water away, electrostatic forces, which attracts the charges, and the van der Waal interactions, which are weak but important. These tiny forces act like glue, maintaining the structure of life. Have you ever noticed how a gecko can stick to walls, or how molecules stay close together without forming strong bonds? The answer lies in a very weak but important force called van der Waals interactions. These are weak attractive forces between atoms or molecules that are very close to each other. They occur due to temporary changes in electron distribution, creating small partial charges. This leads to the formation of temporary dipoles, where one part becomes slightly positive and another slightly negative. These opposite charges attract each other, creating a weak interactions. There are three main types of van der Waals forces: London dispersion forces, which occur between all molecules due to temporary dipoles; dipole dipole interactions, which occur between polar molecules; and included dipole interactions, where a charged molecule induces a dipole in another. These forces are weak, short range, and universal, but their effect can add up. Think of it like Velcro. One hook is weak, but many hooks together create strong attachment. In biology, these interactions help in protein folding, DNA stability, and maintaining cell membranes. In simple terms, van der Waals interactions are weak, short range forces caused by temporary dipoles between nearby molecules. Have you wondered why water has such unique properties, or how DNA stands stay together like zipper? The answer lies in a special type of interaction called hydrogen bonds. A hydrogen bond is a weak attractive force between a hydrogen atom and a highly electronegative atom. It usually involves hydrogen bond to oxygen, nitrogen, or fluorine. In these interactions, hydrogen carries a slight positive charge while the other atom carries a slight negative charge, leading to attraction. There are two main types of hydrogen bonds: intermolecular hydrogen bonds, which occur between different molecules like water molecules, and intramolecular hydrogen bonds, which occurs within the same molecule, helping in maintain its shape. Although each hydrogen bond is weak individually, together they create strong and stable structures. Think of it like a zipper. Each tooth is weak, but many together hold things tightly. In biology, hydrogen bonds are essential for DNA structure, protein folding, and many life processes. In simple terms, a hydrogen bond is a weak electrostatic attraction between a partially positive hydrogen atom and a highly negative electronegative atom. Have you ever noticed how oil forms droplets in water and never mixes, no matter how much you stir? The answer lies in a special phenomena called hydrophobic interactions. Hydrophobic means water fearing. These interactions occur when nonpolar molecules like oil and fats avoid water and come together. In water, molecules prefer to bond with each other through hydrogen bonding, but nonpolar molecules cannot form these bonds. So water pushes them together, causing them to cluster. This leads to formation of droplets like oil in water. Hydrophobic interactions mainly occur in aqueous environment and involve nonpolar molecules. They are driven by water behavior and not direct bonding between molecules. These interactions play a very important role in biology. They help form cell membranes with lipid bilayers. They assist in protein folding by hiding nonpolar parts inside, and they help create stable biological structures. In simple terms, hydrophobic interactions are the tendency of nonpolar molecules to come together in water to minimize contact with it. So remember, hydrophobic interactions help organize life at the molecular level. Have you ever wondered how common salt forms or how opposite charges attract each other? The answer lies in a powerful force called ionic interaction. Ionic interaction are attractive forces between positively and negatively charged ions. They are formed when electrons are transferred from one atom to another. When an atom loses an electron, it becomes a positively charged ion called cation. And when an atom gains an electron, it becomes a negatively charged ion called anion. These opposite charges attract each other, forming an ionic bond. For example, sodium loses one electron to become sodium, and chlorine gains the electron to become chlorine negative. They then combine to form sodium chloride or common salt. In biology, ionic interactions are important for stabilizing protein structure and maintaining enzyme activity. They can also be affected by pH and ionic strength. Think of all it again like magnets. Opposite charges attract while similar charges repel. In simple terms, ionic interactions are electrostatic attraction between oppositely charged ions formed by the transfer of electrons. So, these are the four kinds of stabilizing interaction we have seen so far. So, whenever there is an interaction between molecules, there's definitely a chance of reactions happening in the body. These chemical reactions may disrupt the balance, or there may be sudden changes in the body, or reaction may happen at the slow speed. Anything can happen. So, in order to keep a homeostasis between everything, biophysical chemistry comes into picture. Life follows rules, strict chemical rules. The pH of your body must stay balanced, buffer prevent sudden changes, reaction must happen at the right speed, and energy must be carefully managed according to thermodynamics. Your body is like a perfectly controlled chemical laboratory. Have you ever wondered what makes a lemon taste so, or why our blood has a stable pH? Understanding pH is a key to understanding biology. It is measured on a scale from 0 to 14. pH plays a crucial role in the body. It controls protein structure and affects enzyme activity. Even small changes in pH can disrupt normal biological functions. In our body, different parts have different pH levels. Body has a pH of about 7.4, which is slightly basic and must be tightly regulated for optimal functions. On the other hand, the stomach has a pH of around 2, making it highly acidic. This acidity helps in digesting food and killing harmful bacteria. Concentration means basic. So remember, pH indicates how acidic or basic a solution is, and maintaining the right pH is essential for life. Have you ever wondered how your blood stays stable even when you eat different foods every day? The answer lies in buffers. Buffers are special solutions that resist change in pH. When small amount of acid or base are added, in simple terms, buffer keep the pH stable. But how do buffers work? A buffer contains a weak acid and its conjugate base. For example, carbonic acid and bicarbonate in our blood. When acid is added, the buffer absorbs excess hydrogen ions, and when base is added, it neutralizes it. This helps maintain pH nearly constant. Buffers are extremely important in the body. They maintain a constant pH, which is essential for proper enzyme activity. They also prevent harmful effects of sudden pH changes. For example, human blood pH is tightly regulated between 7.35 and 7.45. Even small changes can be dangerous. In real life, the carbonic acid bicarbonate buffer system keeps your blood pH stable while you eat, exercise, and breathe. In simple terms, a buffer is a solution that resists changes in pH when small amounts of acid or bases are added. Have you ever noticed some reactions happen instantly, like lighting a match, while other takes years, like rusting of iron? This difference is explained by reaction kinetics. Kinetics is the study of how fast a chemical reaction occurs and the factors that affect its speed. In simple terms, it tells us how quickly reactants turns into products. There are several factors that affect reaction rates. Concentration, more particles means more collision, leading to faster reactions. Temperature, higher temperature gives particles more energy, increasing reaction speed. Surface area, smaller pieces provide more area for reaction. And catalysis, substances that speed up reactions without being consumed. But how do reactions actually happen? According to collision theory, molecules must collide, and these collisions must have enough energy called activation energy and proper orientation. Only then does a reaction occur. In real life, cooking happens faster at higher temperature. Digestion is sped by enzymes, and rusting is a very slow reaction. In simple terms, reaction kinetics is the study of the rate of chemical reactions and the factors that influence them. Have you ever wondered why salt is added to icy roads or why antifreeze is used in car radiators? The answer lies in colligative properties. Colligative properties are properties of solutions that depend only on the number of solute particles, not on their nature. In simple terms, more particles means a greater effect. There are four main types of colligative properties. First, vapor pressure lowering. When a solute is added, it reduces the evaporation of the solvent. Second, boiling point elevation. The solution boils at a higher temperature than the pure solvent. Third, freezing point depression. The solute freezes at a lower temperature. This is why salt is used on icy roads to melt ice. And a fourth, osmotic pressure. This involves the movement of solvent through a semi-permeable membrane from low solute concentration to high. It plays an important role in processes like food preservation. In simple terms, colligative properties depend on how many solute particles are present, not what they are. Have you ever wondered how your body gets energy from food, or why some reactions happen on their own while others need energy? The answer lies in thermodynamics. Thermodynamics is a study of energy and how it is transformed. It explains how energy exists in different forms like heat and chemical energy, and how it powers biological processes like metabolism. There are two important laws of thermodynamics. The first law states that energy cannot be created nor destroyed, only transformed from one form to another. The second law states that energy transformation increases disorder, also known as entropy. In reactions, energy changes are important. Exothermic reactions release energy, while endothermic reactions absorb energy. Another key concept is Gibbs free energy or delta G. It determines whether a reaction is spontaneous. If delta G is negative, the reaction occurs on its own, releasing energy. In real life, when you eat food, chemical energy is converted into ATP, which powers movement, growth, and all body functions. In simple terms, thermodynamics is the study of energy, transformation, and their role in life. The most important question: where does energy comes from? When you eat food, your body breaks down glucose. First, glycolysis begins. Then, the mitochondria takes over. Through oxidative phosphorylation, energy is produced. This energy is stored as ATP, the currency of life. Have you ever wondered how your body quickly gets energy from glucose even without oxygen? The answer begins with glycolysis. Glycolysis is the first step of cellular respiration, where glucose is broken down to produce energy. This occurs in the cytoplasm and does not require oxygen, so it is an anaerobic process. In this process, one molecule of glucose, which has 6 carbon, is broken down into two molecules of pyruvate, each with 3 carbons. Glycolysis happens in two main phases. First, the energy investment phase, where two ATP molecules are used. Then, the energy payoff phase, where 4 ATP and 2 NADH are produced. So, overall, glycolysis gives a net gain of 2 ATP and 2 NADH. This process is very important. It provides quick energy, especially during intense exercise when oxygen levels are low. It is also the first step in cellular respiration and supplies important intermediates. In simple terms, glycolysis is an anaerobic breakdown of glucose into pyruvate, producing ATP and NADH. Have you ever wondered how your body carries out energy requiring processes like muscle movement or active transport? The answer lies in coupled reactions. Coupled reactions involve two types of reactions. One releases energy called exergonic reactions and other requires energy called an endergonic reaction. In simple terms, energy from one reaction is used to drive another. But how does this happen? It happens through ATP, the energy currency of the cell. When ATP breaks down into ADP and phosphate, it releases energy. This energy is then transferred to power cellular processes. On their own, endergonic reactions cannot occur. But when they are coupled with the exergonic reactions, they become possible. This energy transfer is what keeps our cell functioning. In real life, coupled reactions are involved in muscle contractions, pumping ions across membranes, and building proteins. In simple terms, coupled reactions link energy releasing and energy requiring processes, making life possible. Have you ever wondered where most of your body energy is actually made? The answer lies inside tiny structures called mitochondria through a process known as oxidative phosphorylation. This is the final step of cellular respiration where most ATP is produced. It occurs in the inner mitochondrial membrane. There are three key components involved. First, the electron transport chain, a series of protein complexes. Second, the proton gradient, where hydrogen ions are pumped across the membrane. And third, ATP synthase, the enzyme that produces ATP. So, how does it work? NADH and FADH donate electrons to the electron transport chain. As electrons move through the chain, energy is released, and this energy is used to pump hydrogen ions, creating a gradient. These hydrogen ions then flow back through ATP synthase, driving the production of ATP. Oxygen plays a crucial role here. It acts as the final electron acceptor, combining with electrons and hydrogen to form water. In simple terms, oxidative phosphorylation is a process where energy from electrons is used to produce ATP, powering your body. Have you ever wondered how energy and molecules are passed from one reaction to another inside your body? This happens through group transfer reactions. Group transfer reactions are processes where a simple chemical group is transferred from one molecule called the donor to the another called the acceptor. In simple terms, one molecule gives a functional group and another receives it. These reactions are very important. They help in energy transfer, drive metabolic pathways, and enable the synthesis and breakdown of molecules. One of the most important molecules involved is ATP. ATP acts as a group donor, especially for phosphates. When ATP breaks down into ADP and phosphate, it transfers energy, helping convert chemical energy into usable forms. These reactions are central to processes like glycolysis, the Krebs cycle, and biosynthesis. In real life, they are involved in muscle contraction, DNA regulation, and fat metabolism. They are also connect in different metabolic pathway, ensuring smooth flow of energy. In simple terms, group transfer reactions move functional group between molecules, helping energy flow through the body. Have you ever wondered how your body converts energy from food into a usable form that powers every movement throughout an heartbeat? The answer lies in biological energy transducers. These are systems in living organisms that convert energy from one form to another. And remember, energy cannot be created or destroyed, only transformed. There are two main types of biological energy transducers: mitochondria, known as the powerhouse of the cell. They convert chemical energy from food into ATP through cellular respiration, and chloroplasts in plant. They convert light energy into chemical energy throughout photosynthesis. In mitochondria, the main process is the oxidative phosphorylation, which produces ATP. This ATP is the energy currency that powers all cellular activities. These energy conversations are essential. They drive metabolism, enable muscle contraction, support active transport, and help in biosynthesis. In simple terms, food is converted into glucose and then into ATP, which your body uses as energy. Think of mitochondria like a generator producing energy for the cell. In simple terms, biological energy transducers convert energy into usable forms to sustain life. Have you ever wondered how reactions inside your body happen so fast, even at normal temperature? The answer lies in catalysis. Catalysis is the process of increasing the rate of chemical reactions using a substance called a catalyst. A catalyst speeds up reactions, but it's not consumed in the process and can be reused again and again. But how does a catalyst work? It lowers the activation energy, the energy required to start a reaction. It also provides an alternative pathway, making the reaction happen faster. In living systems, catalysts are called enzymes. These are highly specific, work under mild conditions, and are essential for lives. Without enzymes, many biological reactions would be slow to sustain life. In real life, enzymes help in digestion of food, and catalysts help break down hydrogen peroxide in cells. These are catalysts, and they are different types, like homogeneous catalysis, and reactants and catalysts are in the same phase. In simple terms, catalysis speeds up chemical reaction by lowering activation energy. So remember, catalysts are the reason life reaction happens efficiently. Enzymes are the biological catalysts. This speeds up reaction. They work like a lock and key, specific and precise. Some enzymes can be regulated, turned on or off. Without enzymes, life would be too slow to exist. Have you ever wondered how fast enzymes work and what controls the speeds of reaction inside your body? That study is called enzyme kinetics. Enzyme kinetics is the study of how enzyme catalyzed reactions occur and how different factors affect their rate. As substrate concentration increases, the reaction rate also increases, but only up to maximum limit called Vmax. At this point, all enzyme active sites are fully occupied and the enzyme is saturated. Another important concept is Km. It represents the substrate concentration at which the reaction rate is half of Bmax. At lower Km means higher affinity between enzyme and substrate. Several factors affect enzyme activity such as temperature, pH, substrate concentration, and inhibitors. There are two main types of inhibition: competitive inhibition, where inhibitor competes with the substrate for the active site, and non competitive inhibition, where the inhibitor binds elsewhere, reducing enzyme activity. In simple terms, enzyme kinetics explain how reaction rate increases, then level off, based on enzyme behavior. Have you ever wondered how your body knows when to speed up or slow down reactions? The answer lies in enzyme regulation. Enzyme regulation is the control of enzyme activity, ensuring reactions happens at the right time in the right amount for the body needs. Cell must maintain balance. Too much activity can be harmful and too little can slow down essential processes. There are different ways enzymes are regulated. One important method is allosteric regulation, where molecules bind to a different site on the enzyme, changing its activity. Another is feedback inhibition, where the final product of a pathway stops the enzyme, preventing overproduction. Enzyme can also be regulated by covalent modification like phosphorylation, which turns enzymes on or off, and through enzyme inhibition, either competitive, where inhibitors compete with the substrate, or non competitive, where they bind elsewhere. Think of it like a tap. When it's open, the reaction speeds up. When it's closed, the reaction slows down. In simple terms, enzyme regulation controls reaction rates to maintain balance in the body. Have you ever wondered how enzymes make reactions happen so fast? The answer lies in the enzyme mechanism. Enzymes work by converting substrates into products through step by step process at the molecular level. First, the substrate binds to the enzyme, forming an enzyme substrate complex. Next, the reaction reaches a high energy transition state where bonds are rearranged. Then, the substrate is converted into the product, forming an enzyme product complex. Finally, the product is released and the enzyme becomes free to work again. One key idea is that enzymes lower activation energy, making reaction happen faster and more efficiently. They do this by bringing molecules together, weakening bonds, and creating a favorable environment for the reaction. There are two main models that explain how enzyme works. The lock and key model, where the substrate fits perfectly into the enzyme, and the induced fit model, where the enzyme changes shape to fit the substrate. Think of it like a machine. Raw material goes in and a finished product comes out. In simple terms, enzyme speeds up reactions by lowering activation energy and guiding substrates into product. Have you ever wondered how reactions inside your body happen so fast, even at normal body temperature? The answer lies in enzyme catalysis. Enzymes are biological catalysts that speeds up chemical reaction in living system. They increase reaction rate without being consumed and can be reused again and again. The key idea behind enzyme catalysis is activation energy. Enzyme lowers the energy barrier needed for a reaction to occur. So reaction can happen quickly, even at body temperature. Without enzymes, reactions are slow and require high energy. But with enzymes, they become fast and efficient. So how do enzymes work? First, the substrate binds to the enzyme. Then the transition state is stabilized. The reaction occurs and finally the product is released, leaving the enzyme free again. Enzyme catalyze reaction by bringing molecules together, orienting substrates correctly, weakening bonds, and creating a favorable microenvironment. There are different types of enzymes catalysis, including acid base, covalent, and metal ion catalyst. For example, enzyme catalase breaks down hydrogen peroxide into water and oxygen, protecting cells from damage. In simple terms, enzyme catalysis speeds up life's reaction by lowering activation energy. Have you ever wondered how the same chemical reaction can happen in different tissues but in slightly different ways? The answer lies in isozymes. Isozymes or isoenzymes are different form of the same enzyme. They catalyze the same reaction but have slightly different structures and properties. This allows them to function efficiently in different tissues of the body. For example, the heart, liver, and muscles and all have different energy needs. And isozymes have meet those specific demands. A key idea is same function but different structure. This helps in fine control of metabolism. There are important examples of isozyme. Lactate dehydrogenase or LDH has different forms that convert lactate to pyruvate, and creatine kinase or CK has different forms in muscle, heart, and brain. Isozymes also have clinical importance. They are used as markers to diagnose conditions like heart attack or liver disease. Think of it like different teachers teaching the same subject but in different style. In simple terms, isozymes are different forms of the same enzyme that perform the same function in different tissues. Proteins are not just stains, they fold into complex shapes. These shapes determine their function, from alpha helices to beta sheets, from primary to quaternary structure. A protein 3D conformation determines how it works. Every fold matters. Even a small change in shape can destroy function. The center of it all is DNA. It stores the instruction for life. Did you know DNA can have different shapes? From aDNA to the common bDNA and even zDNA. Each form has unique structure while RNA stays flexible, helping perform diverse functions in the cell. RNA helps translate instructions into actions. Together, they control everything happening inside your body. Life survives only when conditions stay stable. Temperature and pH must stay balanced for biomolecules to function. Too much change can damage proteins and DNA. That is, too much heat or extreme pH can damage proteins and DNA. DNA is double stranded, while as RNA is single stranded. Why DNA is stable while RNA is fragile? DNA is double stranded while RNA is single stranded. DNA has no 2' OH group while RNA has 2' OH group. So, this makes RNA less stable and DNA more stable. The stability factors involve hydrogen bonds, base stacking, ionic interaction, temperature, pH level, ionic strength, and base composition. If any alteration in these things can affect DNA and its loss of function. So, temperature and pH must stay balanced for biomolecules to function. Too much change can damage proteins and DNA. Too much heat or extreme pH can damage proteins and DNA. At optimal conditions, everything works perfectly. This balance is called homeostasis. Life depends on stability. So, maintaining the right condition is the essential for life. Finally, everything connects through metabolism. Carbohydrates, lipids, proteins, nucleic acids, nucleotides, all are constantly being built and broken down. This continuous cycle is what keeps life going. Have you ever wondered how food you eat turns into energy that powers your body? The answer lies in carbohydrate metabolism. Carbohydrate metabolism is a process by which body breaks down, stores and uses carbohydrates to produce energy. There are three major pathways involved. First, glycolysis, where glucose is broken down into pyruvate, producing a small amount of energy. Next, the Krebs cycle, which occurs in the mitochondria, producing high energy carriers like NADH and FADH2. And finally, the electron transport chain, where maximum ATP is produced. But carbohydrate metabolism is not just about energy, it also involves storage and release. Glycogenesis stores glucose as glycogen. Glycogenolysis breaks glycogen back into glucose. And gluconeogenesis creates new glucose when needed. Blood glucose levels are carefully regulated by hormones. Insulin lowers blood sugar while glucagon increases it. This process is essential. It provides energy for the brain, muscles, and all cells. Think of the food like fuel, metabolism like the engine, and ATP as the energy that powers your body. Have you ever wondered how your body stores energy for long periods and uses it when needed? The answer lies in lipid metabolism. Lipid metabolism is the process by which fats are digested, absorbed, stored, and used for energy. Lipids include triglycerides, fatty acids, and cholesterol. The process begins with digestion and absorption. In the small intestine, fats are broken down into fatty acids and glycerol and transported in the blood as chylomicrons. Next comes transport. Lipid travel through the bloodstream as lipoproteins. For storage, excess energy is converted into triglycerides and stored in adipose tissue. When the body needs energy, lipolysis breaks down stored fats into fatty acids and glycerol. These fatty acids then undergo beta oxidation in the mitochondria, producing acetyl CoA, NADH, and FADH2. This leads to ATP production through the Krebs cycle and electron transport change. Lipid metabolism is very important. It provides long term insulation protection and helps in cell membrane and hormone formation. In simple terms, lipid metabolism stores energy when there is excess and release in when the body needs it. Have you ever wondered what happens to protein after digestion and how your body uses amino acids? The answer lies in amino acids metabolism. Amino acid metabolism is a process by which amino acids are broken down, used, and converted into energy or other molecules. Each amino acid has two parts, an amino group and a carbon skeleton. The metabolism begins with two major processes. First, transamination, where the amino group is transferred from one molecule to another, and second, deamination, where the amino group is removed, producing ammonia. This ammonia is then safely processed in the liver. After removal of the amino group, the carbon skeleton enters different pathways. It can form pyruvate, acetyl CoA, or enter the Krebs cycle. This can be used for energy production, glucose formation, or fat synthesis. Amino acids are classified based on metabolism glucogenic, ketogenic, or both. Amino acid metabolism is very important. It provides energy during fasting and helps in the synthesis of neurotransmitters, hormones, and nucleotides. However, disorders like phenylketonuria can occur due to enzyme defects. In simple terms, amino acid metabolism breaks down protein and converts them into energy and essential biomolecules. How your body builds and recycles the molecules that store genetic information. The answer lies in the nucleic acid metabolism. Nucleic acid metabolism is a process by which nucleotides are synthesized, used, and broken down in the body. Nucleotides are the building blocks of DNA and RNA, and they play essential role in energy transfer like ATP, and coenzymes like NAD and FAD. There are two main pathways for nucleotide synthesis: de novo synthesis, where nucleotides are built from small molecules, and salvage pathway, where the body recycles existing bases and nucleotides. Nucleotides are also classified into purines like adenine and guanine, and pyrimidines like cytosine, thymine, and uracil. When nucleotides are broken down, purines from uric acid and pyrimidines are converted into simpler molecules, and this metabolism is tightly regulated through feedback information and enzyme control. Disorders can occur if this process is disturbed, such as gout due to excess uric acid. In simple terms, nucleic acid metabolism builds, uses, and recycles the molecule that carry genetic information. Why we need vitamins in such small amounts, yet they are essential for life? The answer lies in vitamin metabolism. Vitamin metabolism is the process by which vitamins are absorbed, transported, and stored used in the body. Although vitamins do not provide energy directly, they are crucial for supporting metabolism and enzyme functions. Many vitamins act as coenzymes, helping enzymes carry out vital biochemical reactions. There are two main types of vitamins: water soluble vitamins like B complex and vitamin C, and which are not stored and need regular intake, and fat soluble vitamins like vitamin A, vitamin D, vitamin E, and vitamin K, which can be stored in the body. The process begins with the absorption in the intestine. Then vitamins are transported to the bloodstream, often bound to protein. Their levels are carefully regulated based on diet, absorption, storage, and excretion. Vitamins play many important roles. They act as coenzymes, support bone health, improve vision, aid blood clotting, and functions as antioxidants. But both deficiency and excess of vitamins can be harmful. In simple terms, vitamin metabolism ensures vitamins are properly used to maintain health and support life. Everything in life begins with structure and function. In biochemistry, understanding biomolecules is the key to understanding life itself. We start with the basics: atoms, molecules, and the chemical bonds that hold them together. From there, we explore the composition and structure of essential biomolecules like carbohydrates, lipids, proteins, nucleic acids, and vitamins. But structure alone isn't enough. Stability comes from interaction. Van der Waals forces, hydrogen bonds, electrostatic, and hydrophobic interactions working silently behind the scenes. To truly understand life, we dive into biophysical chemistry, such as pH, buffers, thermodynamics, and reaction kinetics. And then energy. Bioenergetics powers every cell through glycolysis, oxidative phosphorylation, and couple reaction. Enzyme takes control, guiding reactions with precision, speed, and regulation. We uncover the complex unfoldings of protein and the elegant structure of nucleic acids. And finally, everything connects. Metabolism integrates it all, transforming molecules into life, sustaining energy and functions. This is not just chemistry, this is a language of life. Enjoyed the video? Give it a like and share it with someone who loves sciences. Subscribe to Deep Insight, where complex concepts become simple. And do not forget to tap bell icon so you never miss an update, because every video brings you one step closer to truly understanding life. Deep Insight, learn deeper, grow smarter. Thank you.